Hello everyone. Welcome back to the webinar. Today we will talk about recombinant protein DT3C, and we will talk about how can we use the DT3C to perform the antibody internalization assessment, and how can we use the DT3C to accelerate the selection of ADC candidates. So for today's webinar, there will be three parts. The first part, I will talk about the importance of the antibody internalization. And then the second part, we will talk about current internalization assays that on the market. And then the last part, I will introduce our feature product, the recombinant protein DT3C, and how can we use it for the antibody internalization assays. At the very beginning, I still want to introduce the antibody-drug conjugates, ADC. So this is a pretty hot therapeutic in the market. As you see here, we have a ADC structure. The red part here is the monoclonal antibody that can recognize the antigen that proficiently expressed on the tumor cell surface. And we have the blue is the linker, and the yellow part here is the potent cytotoxic, which we usually call payload. So you see that there are three key components that in the ADC. So the monoclonal antibody is linked by the linker and then to the payload. So basically, you see that because the monoclonal antibody that in the ADC, this monoclonal antibody will recognize the antigen on the surface and then binding to the antigen. So after the binding, the ADC will be internalized into the cells, and the antibody portion inside the cells, they will be degraded. And then the manner is usually lysosomal degradation. So after the degradation and the cleavage of the linker, the payload will be released, and the payload will link to the target, so resulting in the cell apoptosis. And at the same time, as you see here, the partial of the payload can be taken up by the neighboring cells, and this process was known as the bystander effect. Currently, there are total 15 ADC in the market. So as you see here, before the 2009, there is only one drug, the Mylotarg, was approved by the FDA. But then from 2010 to 2025, this year, during the past 15 years, they have been already more than 10 ADC were approved by FDA, and they are used for different indications. So you see that the market of the ADC is growing so fast. So the market size of the ADC right now, I think it has been exceeded $10 billion US. And since it's developing so rapid, there are more than 200 types of ADC. They are ongoing the clinical trials. But unfortunately, for this ADC, the active ADC in the clinical trial, I think majority of them, they will fail halfway. However, drawing lessons can still be learned from their clinical termination. And I think these lessons, they will give us a more profound significance for the future ADC field development. Here I will make a summary about 5 key elements that are important for ADC to continuously optimize. So they are target, antibody, linker, payload, and the conjugation. For each of the element, I think I can talk so much about them. And I think for each element, we have a lot of strategies and the lessons that from the past that we can talk about. And for each element, there are still a lot of things that we can keep optimized for the ADC design. So for example, for the target, usually we want the target antigen, they are highly expressed in the tumor cells, and we want the antigens, they are exposed on the cancer cells. For the antibody, usually the specificity and the affinity are pretty important, and we also want the antibody have a very rapid and efficient internalizations. And then for the linker, because the linker is linking the monoclonal antibody and the payload, we want the linker to be stable in the plasma, and then it will lead to efficient release of the payload into the tumor cells. Then for the payload, because based on the previous research, only a very small fraction of the administered dose of the ADC can reach the tumor tissue. So meaning that the payload, we want them to have a very greater cytotoxicity, because in this way, we can achieve a very high or efficient therapeutic efficacy. So we want the payload to be potent. And conjugation technology is another very important element, because it's kind of like affecting the homogeneity of ADC. So this feature is kind of related to the safety of the ADC, because ADC, you see, we have 3 components inside. So sometimes that if the design is not that good, the population will be, the drug population will be heterogeneous, and then resulting in some systematic toxicity. So conjugation technology is another important part that researchers keep doing research. So to have a very better understanding of ADC, so here I show to you some selected FDA-approved ADC in the market. And basically, they can be divided into 2 categories. So the top 6 here, they are used for the hematological tumors. And the bottom here, they are for the solid tumor treatment. So because of the time we are limited today, I'm not going to introduce all the ADC drugs that in the market. I want to highlight some of the features of ADC to give you a better understanding. So the first is that if you take a look at the target here, you see that different ADC, they have a different target, and definitely for the liquid, for the blood tumors, you see that the antigen, they usually come from the CD series, like CD33, CD30. And for the solid tumor, it looks like that the antigens are pretty diverse, and then to target are pretty diverse. And HER2 seems to be a popular and a matching target. And some of the target that are pretty novel, like the Nectin-4 and the TR-alpha. And another thing is the number. Did you see the number here? This is the DAR value. So DAR value is a key property that used to measure the quality of ADC, because the DAR value can significantly affect the ADC efficacy. It's essential to measure the DAR, because the low DAR value sometimes indicates decreased efficacy, and a relative high DAR value may also negatively impact the safety. So overall, I would say that ADC design is a kind of like a balance between the efficacy and the safety. So there are 2 ADC that I want to highlight in today's webinar. So the first one is the T-DXd. It's come from the AstraZeneca and the Daiichi Sankyo. This ADC at the very beginning is used for the treatment of HER2 positive breast cancer. And then recent years, the indication has been expanded to other solid tumors. So 2 features about this T-DXd ADC. The first is the DAR. See, the DAR value here is 8. It's meaning that for each antibody is carrying 8 payload. And also the payload is a pretty novel payload. It's called DXd. DXd is a pretty strong payload, and I think the bystander effect for this payload is pretty good. Another drug that I want to highlight in today's webinar is this one, the Padcev. This drug is produced and marketed by a cooperation between the Astellas and the Seagen. So it's the first in-class therapeutic indicated for the treatment of the Nectin-4 positive urothelial cancer. And the linker used in this ADC is dipeptide linker. Payload is not that novel. It's a pretty traditional one, the MMAE, but the highlight part in this is the target. So Nectin-4 is a pretty novel target. In all, you see that the ADC in the market, they have some similarities and also they have difference. But we are so glad to see the expanding of the antigens, and we are pretty happy to see the advancement development of the linker technology and the payload novelty, because all this, they will bring a bright future for the next generation of ADC. So come back to today's topic. I want to focus on the internalization efficiency of ADC. So this one is a key feature or a challenge that in the development of ADC. So if you take a look at the right panel that I showed you here, this is about a mechanism of action of internalizing ADC. So you see that after the ADC is internalized with the degradation, no matter early or late endosome or the lysosome, the payload expected to be fully released, right? And the internalization rate and the kinetics of endosomal lysosomal trafficking upon the tumor antigen binding to the ADC can directly influence the efficient payload released, and as well as the cancer cells killing. So early studies have already showed that there's an intricate correlation between antibody internalization and the tissue penetration. Right now, most of the current ADC, they are designed against the target antigen that have a very pretty good efficient internalization, because this can facilitate the ADC entry upon the recognition. However, regarding the internalization efficiency, many factors can affect this one. So like the antibody affinity and the specificity, and also the antibody types and subtypes, isotypes can affect the internalization efficiency. So for example, the IgG1 and IgG2, they may have different internalization efficiency. And also the cell types and the extracellular environment factors that around the cells can also affect the internalization efficiency. So meaning that during the ADC design, the internalization assessment will be a very important part during the design. And inefficient internalization not only lead to efficient efficacy, but also will lead to the off-target release of the ADC. So therefore, it's resulting in a systematic toxicity. So safety and efficacy both will be affected by the low internalization efficiency. So then talking about the internalization, we need to understand the endocytosis pathways of ADC. So based on the research, we understand or we know that there are three main endocytosis pathway of ADC that in the ADC here. So the first one is the castration-mediated endocytosis here. And the second one is the Cabli-mediated endocytosis. And the third one is the micropincytosis. The first two, they are regarded as the receptor-mediated endocytosis. And the third one is a receptor-independent endocytosis. So I would say that the first one, the CME pathway, is regarded as the most important endocytosis pathway for a majority of ADC right now. And this one is pretty complex. It varies significantly, depending on the different cell types, different receptor ligand pairs, and also the serial contact. So even the same receptor or the ligand, they may have different behaviors, such as the varying binding affinities and different internalization efficiencies, meaning that we probably need to spend more time to do more research. And also, if we go back to the target receptor for the ADC discovery, here is a summary of the ADC target that from the active clinical trials. So I summarize the top 10 targets that you will find in the active clinical trials. So you see some familiar target like the HER2, Nectin-4, c-MET, EGFR, HER3. So we found that most of the target or most of the company right now, they focus on the mature and the validated target. So based on this target layout, I would say that they concentrate on the mature target, mainly those that have been already being approved by the ADC drugs or have been validated in some other therapeutic approach. I think one reason here is because that there are enough research or partial research that have been done about the endocytosis pathway for this target. So researchers know that how can they design the ADC better because they know a lot about them. And then for the continue here is a summary about the endocytosis pathway that from the research showing that some of the target, like which endocytosis pathway they have. So for example, for majority of them, so as I mentioned before, the CME is the most common and the best studied pathway in the eukaryotic cells. So as what you see here, see that the TROP2 and the BCMA, they have the CME pathway. And for the Nectin-4, the novel target, it seems not. It's a receptor-independent pathway. And the HER2 seems to be a pretty complex one. So the endocytosis mechanism for HER2 is pretty diverse. It's including the CME, but also the cabinet-mediated, and also the click and the JAK. So what I want to highlight here is that understanding the receptor-ligand interaction and their role in the endocytosis can give us a great potential for the future clinical application, particularly in knowing the disease mechanism and also to design the next generation of ADC better. So overall, I think the study about the endocytosis is pretty important in the future. So I'll come back to the second part for today's webinar. I will introduce the current internalization assessment tools, because we already talked about how important is the antibody internalization. And actually various tools have been developed to facilitate the ADC discovery. The first one is an assessment tool based on the pH-sensitive probes. So because these probes, we can make a detection about the fluorescent signal change via the flow cytometry or fluorescent microscope. And the second one is a pretty directly observation about the ADC distribution in the cells. It's an image-based tools. And the third one is the toxin conjugated by performing the cell linking assay, we can indirectly make an assessment about the internalization. I will introduce them one by one. So the first method. This method is using a highly hydrophilic and the bright pH sensor dye. So it's called the pH sensitive probes. So this probe will conjugate to the fat fragments of the IgG, and the probe is not sensitive, is not frozen at the neutral pH. So outside the cells, because the pH is usually around 7, so it's a neutral pH, and the probe is not frozen. But when the antibody is getting inside to the cells, the pH inside will decrease, so the probe will become highly frozen at this environment. So overall, the conjugate will be minimally frozen at the neutral pH and will become brightly frozen upon internalization into the cells. So by this signal change, the frozen signal change, we can use the flow cytometry or the frozen microscope to study the mechanism and the regulation of the antibody internalization. So this is the first one. Then the second one is a pretty straightforward observation. So as you see here, with the confocal microscope or some compatible imaging software, we can perform the assay in a real-time manner. You can directly see the co-localization of ADC with the cell organelle. It's kind of like you can clearly see the position of the antibodies at different time points. You can also, based on some of the imaging systems, you can also analyze the data, correct the signal distribution. You can correct the data and perform the analysis. But for this one, you probably need some imaging instrument. Then the third internalization assessment tool is immunotoxin-based assay. So this one can be performed in a high-throughput manner. It relies on you need to form the immunotoxin. So with the molecular antibody and also the toxin. So after the immunotoxin is internalized into the cells, the toxin will be released inside the cells and it will induce the cell toxicity. So by evaluating that the survival status of the cells, you can kind of make assessment about the internalization efficiency. So what I showed here, the apoptosis behind here is that because the toxin can be released and it will lead to the ADP ribosylation of the active EF2 and then causing the cytotoxicity by inhibiting the protein translation mechanism. So overall, the third internalization as this assay can be processed in high-throughput manner. So we can usually use it for the initial and the primary screening. If you have so many candidates, you can use this tools to perform the initial screening and get several optimal candidates to move on to the next experiment. And for this part, I will stay at here because for the third type of the assay, there are two important tools. The first one is the recombinant DT3C from our company, and the second one is MAB-ZAP. So you see that for both of this one, they have the toxin. So for recombinant DT3C, we have the diffusory toxin, and the MAB-ZAP, we have the cyparine. So cyparine is conjugated to the secondary antibody, and this secondary antibody will bind to the primary antibody, and the primary antibody will recognize the antigen on the surface. But for DT3C, you do not need to have the secondary antibody because the 3C domain from the streptococcus protein G, because this 3C domain, it can recognize the assay of the IgG. So it can bind to the IgG from different species. It's pretty, it can be applied pretty wide. But overall, these two, they are pretty popular in the market. Before I move on to our feature product, recombinant DT3C, here I did a simple comparison between all three types of the tools. So the principle for the first one is that you have the antibody, you label them with the pH-sensitive probes, and these probes, they are non-fluorescent at the neutral pH, but will have a bright fluorescent upon the internalization into the cells because the pH decreased. And with the flow cytometry and or the fluorescent microscope, you can measure the signal change from the fluorescent signal change. And the feature for this type of assay is that it can be pretty high specific. And also the assay can be quick, the efficiency can be quantified. For the second one, the feature is that you can do a real-time observation and the resolution can be high. You can directly see the co-localization of the ADC and the organism-specific probes. But you probably need the confocal microscope and some compatible imaging software. For the third one, we also rely on the toxin. So the toxin will conjugate it to the antibody. And after the immunotoxin internalized into the cells, the toxin is released and internalization efficiency can be indirectly evaluated by the cytotoxicity. This one, the feature is that you can perform the assay in a high-throughput manner, and it's very good to use this assay for our primary and initial screening. And also the method is quite easy. You can use different types of the cytotoxic assays to make the assessment. Okay, finally, let's come back to our recombinant DT3C. So what is the recombinant of DT3C? You see that we have a diffuse toxin without the receptor binding domain, and we link it to the 3C domain of the Streptococcus protein G. So this is a recombinant protein. DT3C is kind of like a mimic the ADC internalization. So DT3C first will bind to the Fc region of the IgG, and after this conjugate internalized into the cells, it will cause the cells to die. And by performing the cytotoxic assays, we can make assessment about how many immunotoxin has been internalized into the cells. So comparing to the MAB-ZAP, DT3C is pretty small. You do not need to have a secondary antibody, and you do not need to worry about the species of your secondary antibody. It's pretty, you can spend low cost and perform a primary screening. And also because the 3C domain of the Streptococcus protein G, so DT3C can form the immunotoxin with IgG from different species. So it has a wider application comparing to the MAB-ZAP. Our DT3C has a verified high purity. So we guarantee that the purity will be higher than 90%. And the data is come from the SDS page and the HPLC. Here is a general workflow to show you how we can use the DT3C to make the assessment. And then later I will also show you a short videos. So overall, the assays will first, you mix the antibody and the DT3C to form immunotoxin. So this process is pretty quick. You just mix the antibody with the DT3C and perform the 30-minute incubation at room temperature of 37 degrees. Then they will form the immunoconjugate, right? And at the same time or before this step, you can plate the cells. So you prepare the cells that you want to make the assessment. Then you count the cells and dilute the cells and plate the cells to the 96 well plate. And then the next step is that you mix the immunoconjugate with the cells and perform another round of incubation. Then the last step will be that you perform the cell cytotoxic assays to correct the data and have the data analysis. So the whole experiment is easy to prepare and also it's very convenient. You can just use the 96 well plate to perform the assay in a high-throughput manners. And MTT cell preparation and some other cell cytotoxic experiment can help you to measure the the cell metabolic activities. So it's kind of like indicator of the cell availability, proliferation, and cytotoxicity. So it's quite easy. Okay, as I mentioned, we have a short video that I can show you to, from our YouTube channel. So based on this video, you can have a direct view about how you can do the assay. Okay, so the first step will be preparing the cells. The cells we are using here is the SKBR3. So this one is the endogenous cell line. So our lab members, they are doing the digestion of the cells and crack the cells, and then later they will do the counting. So this step is just, the cell preparation. It may take some time. So right now, our lab member is remove the cells to the flow tube, and then he or she will use the flow cytometry to count the cells. So we use the flow cytometry to do the cell counting. And then after we got the number of the cells, we will perform the dilution, make sure that for each well, the cell number will be equal. So then we will put it back to the incubator for the overnight culture. And then for this step is the preparation, between the DT3C and the antibody to form the conjugate. So after we have the mixture, we will, here's the 30-minute incubation that I mentioned before. So after 30-minute incubation, the conjugate will form. And here we add the conjugate to the cells that we prepared before. So after the incubation between the conjugate and the cells, the last assay that we performed here is the MTT assay. So this is the cell cytotoxic assay. It's a pretty common experiment that you can find in the lab. Then we can use the microplate radar to correct the data, and then later we will get, like the detection result. Like here, we have the DT3C group, and then we have a conjugate group, and you will see a sharp decrease in the cells, the survival of the cells for the conjugate group. If you want to take more about this video, we have one in the YouTube channel. Yeah, you can check, keep looking at this one if you have questions. Okay, so here there's another application scenario from the literature. DT3C can also be used in combination with the hybridoma technology to facilitate the screening process for good. You can spend more time in the screening stage and then save some more time in the later. So like what I showed here in the literature, the supernatant was from the hybridoma library, work pre-incubated with the engineered toxin DT3C. So internalization assessment with the supernatant from the hybridoma. So early process, and in the first stage screening, then later what they got is a monoclonal antibody with excellent internalization capacity for the future ADC design. So meaning that our DT3C can be used in a lot of different applications. So then the last part here, I want to highlight the features of our DT3C. So the first is that we prepare the conjugate in a very easy way. So the monoclonal antibody and the DT3C, they will be formed like a 30-minute incubation at room temperature or 37 degrees. And because the 3C domain from our DT3C, so the DT3C can bind to IgG from any species, like the human, mouse, rabbit, goat, to form the conjugate. And also the assay will be easy. You can use the MTT, WST1, or the CTG assay to make the assessment, to perform the cytotoxic assay and then to get the efficiency result. And also based on our validated result, the conjugate will reduce the cell availabilities only when they are internalized by the target cells. So as I showed you here in the right panel, you see that in this validation result, we have the DT3C group and we have the monoclonal antibody only group, the positive antibody group. You see that there's no significant reduced cell availability was observed. Only the conjugate is internalized by the target cells. You see a sharp decrease in the cell availabilities. And also, we have already done different bioactive assays for different cell lines. So as I was showed here, from the SKBR3 to the MCF7, all these cell lines, they are the endogenous cancer cell lines. So we did the DT3C killing assay with all the cell lines, and we see pretty good result for some of them. And also we have some assays with the cell lines that overexpress some of the targets like the CCR8, DLL3. So for this overexpression cell lines, we also perform the DT3C with this and we see good result. So this just for your reference. And if you want to know more about some of the data, you can contact us and we can talk more here. So for today's webinar, the take-home message here is that DT3C is a simple, efficient, and economic tool to evaluate the efficiency of antibody internalization, and it can facilitate the discovery of ADC drugs. So lastly, please give me several more minutes to introduce our company. We are CUSABIO, meaning customer satisfied biological reagent, and we are specialized in transmembrane protein expression and antibody screening. We have 5 different product lines. We have the ELISA kit, proteins, antibodies, molecular biology products, and also the small molecular antigens. Our product has been exported to more than 80 countries around the world, including Europe, America, Japan, and Germany. Also, we have a very good cooperation with famous companies like the Pfizer, Sanofi, Bayer, Johnson & Johnson, Moderna. Our product has been recognized by these famous companies, and we have a very good relationship with them. So last, thank you for your attention here. And if you want to stay connected with us, please scan this barcode. This is a linking barcode. If you have more questions or you want to communicate with us, we can talk more there. Yeah, thank you so much.
Thank you, Thank you for the wonderful sharing from Dr. Zhou. I have some questions that I would like to discuss with you. Also, if any of you have questions or would like to communicate with us, please feel free to post them in the chat window. The first question here is that, what is the effective concentration range of DT3C?
Okay, so based on our result, the effective concentration range can be 1 to 4 micrograms per ml. And but we still suggest our users to determine the optimal DT3C themselves, because I know that different labs, they have different experiments. So the strategy here is that you can determine the optimal DT3C concentration first. So you can establish the DT3C concentration gradient. And in this step, you can keep the antibody concentration fixed. So once the optimal DT3C concentration is fixed, then you can use the same method to determine the optimal antibody concentration. So then finally, you have the optimal DT3C concentration and the optimal antibody concentration. Then you can perform the experiment to detect the antibody internalization efficiency based on different dilution ratios.